So the worms that we use are a mutant type of worm. Again, these ones are actually more like X-Men, but like if anyone's seen Sky High, the guy that glows in the dark, maybe this is that. So the SED1 gene is the execution gene for apoptosis. So when cells are undergoing death, the cells that start to phagocytize, which means eat them, will exhibit this. So when we look at this under the microscope, you'll see cells, smaller ones, surrounding the larger cells that they're consuming. And so what happens though is we have a strain where this gene is bonded to the GFP gene, which is green fluorescence protein. Wow, I can't say words today, it's bad. Okay, but I don't know if you've probably done labs in science classes before. Like this comes from jellyfish and a bunch of other organisms. But by binding this, whenever the SED1 gene is active, those cells glow. And so under the microscope, we can actually see green glowing dots of the cells that are killing other cells during apoptosis. So what's happening here is this is a C. elegans under the fluorescence scope. It's a really wonderful piece of equipment. But these are the gonads, these loops. So C. elegans are hermaphroditic, so they grow their own eggs and they develop their own sperm, and then they fertilize within themselves, and then they just lay fertilized eggs, which is why they're, again, really good research models. We don't need to mate them. But what's happening is as the eggs develop, as anything develops, cells are constantly growing and being killed to form limbs and organs and the specific structures of the body. And so this is background apoptosis. This is happening all the time. All these cells are constantly undergoing apoptosis. So that's why I brought this picture. I wanted to just show you what it would look like on a normal cell. But the fact is, we can see that apoptosis is happening because we're using this mutant strain of worms. But this is the overall pathway of apoptosis in C. elegans, if you're interested. I am. So the experimental design is as such. We would use these worms, we'll grow them on a plate until they're adults. Then we would take the worms and plate them onto different plates. When I say plates, they're like bacteria plates, agar. So we'd grow bacteria on the agar to feed them, or not feed them if we're putting them in the dower. And then we would put these under a UV radiation machine and radiate them at 20 millijoules and 50 millijoules, and also at 0, which means we just didn't radiate them. We would let them sit for a day. So we would do this on, I'm going to talk about when we do this on the next slide, but we do this one day, the next day they'd be radiated, and then I would come in a day after and analyze what happens. So I can count the apoptosis incidence under the microscope by looking. It's really hard, and I'll talk about that in a second. And we also did a functional assay where we measured their fecundity, which means like their ability to breed. And so we found we were checking to see if they would lay the same amount of eggs and if the same amount of eggs would reach maturity in these worms after they've been radiated. They didn't, spoiler. So a typical week in the lab is Tuesday. I would set this experiment up. We would grow them. Wednesday, they would be irradiated under the UV machine. Again, thank you for Dr. Creaser for letting us use that piece of equipment. It was very, very helpful, because initially we had started out thinking about using genotoxic stressors like chemotherapies, but UV radiation is the same kind of process, and that was a much more feasible experiment. And then Thursday, I would do the analysis. I would count the worms that hatched from the eggs that I had counted, and then I would go into the microscopy lab and check to see all of the apoptosis on the scope. So we found that apoptosis actually increases with radiation exposure. So if you look here, this worm has had no radiation, it's at 0, and the only thing that I could kind of assume was a regulated UV radiation caused apoptosis is that small circle. And so that's not always the case, because under the microscope I had to focus up and down the entire time, because if it was a circle, it means it wasn't resolved enough. So I would go in further to see what it actually was, and it would either expand into the ring that I was looking for, or it would be just other incidents of background radiation. And so when I'm saying like the rings I'm looking for, they're specifically caused by the genetic damage they receive, either radiation or just generally, because all of the other green you see, the lighter green that doesn't seem as pronounced, is just background apoptosis that the cells are undergoing to keep a maintenance of the body. But over here, I'm going to go in further. It's hard to see, I have a zoomed in picture, but there's small, very, very defined circles where cells are being consumed as they're dying. But I wanted to point out what was really cool. Do you see these large green splotches right here? Those are eggs. And so the eggs are actually undergoing apoptosis as the baby worms inside develop. So it's really cool. So you can actually see, like, sometimes, yeah, these are also eggs. So these ones are already laid, so they're gonna have, they wouldn't hatch because they were on a slide and they were all paralyzed with lobamisol, but they would normally. So, Anyway, this was only 20 microjoules, by the way. Like to see this stark increase. It's millijoules, not microjoules. I knew I was going to mess that up once today. So we got out of the way. So this is how I quantified apoptosis. This is actually the tail end of the 20 millijoule picture you saw before. So if you look right here, this is a really, really pronounced circular structure. So the green that's around that, I know the whole thing's green, but you see like that more pronounced green is actually cells surrounding a cell as it's being apoptosized. There's more here, they're less pronounced. Again, that would be something I had to fiddle with under the microscope to actually look for, and it's kind of tedious work. I know it's staring at worms, but it got tiring. But as you can see, like we didn't see many of these structures in the 0 millijoule worm. So over here is I have now, these are my results. Again, a lot of this is preliminary, and I want to do all of this again with much larger sample sizes, but this graph itself is kind of indicative of the fact that this model is working. So if you look at the bars, these were incidents of apoptosis as radiation increased. So we had a lot more cell death at 50 millijoules than we did at 20, which was more than 0. And so if you look at the left, I have p-values. The difference between 0 millijoules and 20 millijoules is 0.046, which is under the threshold of 0.05. And if you don't know what a p-value, that's okay. It's a statistical significance, which means that my results are only 4% likely due to random chance of 6%, if that makes sense. So a p-value basically says, yeah, this isn't random. If it's higher, it's worse. You want it low as possible. So the difference between 0 and 50 is a much lower number, and that's much more indicative that the radiation is actually affecting the amount of apoptosis, which makes perfect sense as it causes genetic damage. Again, though, preliminary, need to do it again. And if we look here, so I did do a Dower experiment towards the end of the semester. I didn't have time to complete this one. We honestly were surprised that we got this far. We were still trying to develop the model, but basically what we have here is we have Dower at 0 microjoules, zero millijoules versus 50 millijoules in Dower. And so if you look, there's not really a stark difference, but 50 millijoules is still affecting the Dower worms. But if you look at the non-Dower worms, that percent difference between apoptosis incidence is staggering. So I thought it was really funny that the amount of apoptosis in a worm that had not been radiated but didn't go into Dower was as high as a Dower worm that had 50 millijoules of radiation. So I know millijoules doesn't sound that much. If we looked at these worms on the microscope, they looked awful. They looked like they were starting to just die. The eggs that they were laying were mouse-shaped. I felt awful doing this experiment. It's very important, but like, I just didn't like that. So, anyway, so this is the results of the egg-laying assay. If you look here, this is the amount of eggs laid, and then the blue is the amount of worms hatched from those eggs. So as we increased radiation, the egg concentration decreased along with the worms hatched concentration. And then we compared these results to Dower to see if the fecundity was affected. So worms that went into Dower that experienced radiation, blue is 0 and orange is 50 millijoules of radiation, they still hatched and laid more eggs and worms than any of the others. And so it seems like this stress resistance, even in scenarios in which they did not experience actual variable radiation, they were still able to breed better because that stress resistance conferred into their fecundity. So future experiments, we're going to repeat the Dower experiments, like I said, because those are really interesting and they're kind of the focus right now to see if this model is worth continuing. So I want to do those with larger sample sizes. We're also going to use DAF mutant strain worms, which these worms either go into Dower or don't go into Dower. And it's a consistent, they're mutated to do that as such, and they're going to be really good drug targets for testing to see whether or not this will work in this scenario, whether the Dower that they go into due to the scenarios is what's affecting how the treatment is working. And so the Dower mutant worms are exactly like the graboids from Tremor mutant worms, except nothing at all like those in any way. So I love this movie. So we're also going to be looking at radio modulating agents. I almost said sensitizing, but it kind of does the same thing. So these are drugs that will either make cells more prone to damage by radiation or less prone. And these kind of drugs would be the perfect thing to test on these things. And so they would sensitize cells the same way that Will Ferrell was sensitized during Night at the Roxbury during the Vegas. Again, a lot of 80s references, I apologize. It's your fault. But these are my references. You always cite your sources, or the Incredible Hulk will beat you up. I have acknowledgements. I'd like to thank everyone for coming, obviously. Thank you to Dr. Eric Williams for putting up with me this whole semester and helping me throughout the entire process. Thank you to Dr. Creaser for being my journal advisor and also offering your input and equipment for this. Nicole and also Benji, who were also students working with Dr. Williams this semester. They offered a lot of input and were very helpful through the entire process. I'd like to thank, obviously, the university and yourself, Dr. Buell, for helping me for the honors thesis. And as long as it's everyone, yeah. And I'd like to thank everyone for coming to my science party. Thank you. Now is the hard part.
Oh, questions, comments, suggestions for Kyle?
Yes,
So, what do you anticipate people being able to build upon your experiments once you're done? This is laying the groundwork for future experiments. What are those, and how do you see those going?
So if this model is worth continuing, like I said in the future experiment slide, they can use these radiomodulating drugs to test on them, to see if these drugs are actually making the cells more prone to the radiation or less prone. Because if we can figure that out, those kinds of drugs can then be used in humans to mitigate the effects of the negative
And those
treatments.
drugs would target like the healthy cells to protect them during radiation.
Yeah, that would be the hard part, specifying
Right.
targeting healthy versus cancer.
Yeah.
They are different, but
Right.
not very, which is why
Well,
your
that's why
immune
we have the
system
effective
still know.
radiation.
Yeah, exactly. Yes.
So you talked about, well, you're looking at the difference between Dower and non-Dower worms.
Yeah.
Have they done experiments looking like RNA-seq data for Dower versus like healthy?
So I have looked at some papers that didn't look at RE-seq data for Dower, but for other, where we spent the entirety of your cancer genomics class doing RE-seq on different papers. But from what we noticed, we didn't find a ton of data specifically on this, although that is definitely something that I think would be worth a follow-up if we can see the difference expressions in genes and see like what genes are more active in Dower versus that might be the exact, like that would be something I think should have been in the future experiment slide, where after we figure out if the model's worth following, then start to look at the genes that vary between the two, because that obviously is kind of what the whole point is.
Other
That's a great idea.
questions, comments, and suggestions?
It looks like this project was quite related to the cancer genomics class that
Yes.
you took. So
No, yeah,
there
so-
was a lot of synergy between them.
Yeah, it was really funny actually, because I came up with this research project over the summer to do for my thesis, actually a little bit before that, and then as we learned in cancer genomics, the poster session that we held yesterday, my poster had a lot of data that actually seemed to correlate, and it was also about chemoresistance. That's one of my favorite things about being a student here in the science department, is your classes just start to build on one another, and everything is just related, and it's really awesome.
I would point out that you developed this project completely on your own. Like normally when there's this kind of a science project, I would have the student work on my project, which is muscular dystrophy, but this was such a good idea that Kai was able to develop it all completely on his own.
Other questions, suggestions, or thoughts for Kyle as he heads into his next semester? No. You're welcome. Well, thank you so much, Kyle. I want to be part of everyone's projects. Please eat more food.